Hi everyone, welcome to today's product training. I'm glad to have you here. Today we are going to introduce our professional solution for nucleic acid contamination, brought to you by Scavenger Longlight technology. Let's take a quick look at what we'll cover today. We'll start with the background of the contamination issue, then talk about where the contamination comes from, the limitations of traditional methods. After that, I'll walk you through our new product solution, its core advantage is performance data, how it compares to competitors, and finally, the usage instructions and some real cases. First, let's talk about the background. In molecular labs, techniques like PCR, Parker sequencing, they all rely heavily on pure nucleic acids. But here's the problem. Even trace amounts of DNA or RNA contamination can be amplified exponentially during your experiments. That leads to false positive results, unreliable data, or even total experiment failure. That's a huge waste of your time and resources. This nucleic acid contamination has become a hidden, pervasive threat that every lab has to face. So where does this contamination come from? First, cross-contamination between samples. This is very common. It spreads through shared tools, pipette tips, or even aerosols during operations. Second, reagent contamination. If your reagents get contaminated by accident, every single experiment using that reagent will fail. That's a big problem. There are two more critical sources. One is amplicon contamination. The high concentration amplicons from PCR reactions are extremely dangerous. Once they form aerosols, they can spread all over your lab. The other is plasmid or standard contamination. Those high concentration positive controls, if they leak, they can become persistent contamination sources that are hard to get rid of. Now you might be wondering, what about the traditional solutions? That's why we developed our next-generation nucleic acid scavenger. This product provides comprehensive protection for your lab environment and equipment. It can quickly and safely remove DNA and RNA contamination from surfaces, equipment, consumables, even the air, completely eliminating those false positives. It works through a dual mechanism, degrading DNA and RNA rapidly in a non-sequence specific way, eliminating contamination at the root. Let's look at its core advantages. First, it has broad applicability. It works on benchtops, PCR machines, biosafety cabinets, pipettes, centrifuge tubes, tips, basically every scenario in your molecular lab. Second, the performance is outstanding. Third-party tests verified that it achieves over 99.9% removal rate for both DNA and RNA, ensuring your experiments are accurate and reliable. It's also incredibly simple and fast to use. No complex procedures, just spray it on the surface, wait 5 to 10 minutes, then wipe it off or let it air dry. It really improves your work efficiency, and it's totally safe and non-toxic. It has neutral pH, non-corrosive, odorless. It's safe for you and the environment, and at one tadmage your expensive equipment or consumables. Now let's look at the experimental data that proves its performance. First, the gel electrophoresis experiment. As you can see, after treatment with our scavenger, the nucleic acid bands completely disappeared, showing effective removal. Then we did PCR detection verification. The positive control had a clear band, but after our treatment, the signal was almost gone. We also tested it with different sample volumes. No matter how much nucleic acid we put in, from 200 Ang down to 40 Ang, our scavenger degraded almost all of it, even with just 3 minutes of reaction time. That's really powerful. We also did a metal corrosion test, following the national standard. The result confirmed that our product has essentially no corrosive effect on common metal surfaces. And we tested its compatibility with different materials. We applied the Scavenger on various surfaces, waited 20 minutes, then cleaned it. As you can see, it works perfectly with TIFI, instrument-painted casings, pipettes, and ABS plastic, no damage at all. It also works great with other materials like PEEK plastic, pipette tip boxes, and aluminum alloy, so you don't have to worry about it damaging your lab supplies or instruments at all. When we compared our product with competitors, we found that our Nucleic acid Scavenger outperforms others across all dimensions. It has significant advantages in performance, safety, and cost-effectiveness. Now let's talk about how to use it for regular surfaces like reaction wells, benchtops, or instrument surfaces. It's very easy. First, braid even lie to cover the whole target area. Then let it sit for 5 to 10 minutes. Keep the surface moist so the Scavenger can work. Finally, just wipe it clean with a paper towel, or rinse with water and let it dry. Remember to wear gloves to avoid direct contact with your skin. For smaller parts like piter components, you can soak or rinse them with the product. Just soak them for 10 to 15 minutes, then rinse thoroughly with sterile water and air dry. A few notes: don't mix it with strong oxidizers. Also, the product is light yellow. The color might darken a bit after long storage, but that doesn't affect its performance at all. Alright, that's all for today's training. Thank you for your attention.